Good afternoon, and welcome to today's Congressional Neuroscience Caucus briefing, celebrating 75 years of scientific discovery and invention for the National Institute of Neurology and
Psychiatry. These are the powers.
NINDS. You know, I'm still struggling with that. Okay, so I'm Mark Resnick, and I am, my day job is professor of physiology, biophysics, and psychiatry at the University of Illinois College of Medicine, but that's not why I'm here. I'm chair of the American Brain Coalition, and the American Brain Coalition, in partnership with the American Academy of Neurology, the American Neurological Association, the American Stroke Association, Dana Foundation, Society for Neuroscience, the Association for Frontotemporal Degeneration, the Kavli Foundation, Eli Lilly, Lundbeck, Mitsubishi, Tanabe, Novartis, and Neurocrine Biosciences. Don't think I left anyone out. Is proud to present, to host today's briefing alongside the Congressional Neuroscience Caucus. So ABC is thrilled to work with the Congressional Neuroscience Caucus, which is co-chaired by Congressmen Morgan Luttrell and Mike Thompson. And the Congressional Neuroscience Caucus was formed in 2010 to raise awareness of the essential role of brain research and what it plays, of the essential role brain research plays in human health and society, and highlight the progress and promise of neuroscience research and promote federal policy that supports continued innovation in the field and the resources to continue that innovation. Both members of Congress, both Congressmen Luttrell and Thompson, are excited to attend and will be popping in during the briefing, and they're not here yet. And when they come, we will finish one question and then allow the members to speak. But we have a special treat. We have Congressman George Whitesides here from California, and he'd love to say a couple of words. Congressman.
Thanks, everyone. I'll be brief. I'm George Whitesides. I'm a new congressman from the north side of Los Angeles County. I am on the science committee, and I just think that the work that you all are doing is so important. And I just want to say thanks to all of you in this community for what you are doing for folks. When I was young, no kidding, I'll tell you a quick story. When I was young, I graduated from college in 1996, and I called up a good friend of my father. My father was a professor of chemistry at Harvard for many years, very involved in the biotech industry, and I called up a friend of my father. He was the head of the CIA at the time, and I said, I think about three things to do, and this, no kidding, three things that I'd like to do in my life. Either one is the internet, 1996. Second thing is space. And the third thing is neuroscience, no joke. And this guy, John Deutsch, he says, well, I don't know much about the internet, could be interesting, you know. Neuroscience, really fascinating, sort of like at the time, you know, the early days of biotech, you know, the tools are being developed, and it sounds like a great choice. Whatever you do, don't go into space. Because nothing interesting is happening. I did not listen to him, and I spent my entire career in space. You know, I became the chief of staff at NASA and ran Virgin Galactic and various other things. Anyway, the point is that I'm super fascinated in the work that you all are doing. Very good friends with Tom Khalil and work closely with him in the Obama administration, who was, of course, one of the folks who helped do the BRAIN initiative. And whatever I can do from my perch in the science committee, or to just talk to other members of Congress to advocate for the work that you're doing, consider me a friend and an ally, and I'm just so grateful to be able to listen in for a bit today. Thanks very much.
Thank you.
The question I just asked the congressman was, so you really are a rocket scientist? And we're just neuroscientists. But I'd like to introduce one of the chief neuroscientists right now, and that's Dr. Walter Koroshetz. who's director of the National Institute of Neurological Disorders and Stroke. And Dr. Korshitz joined NINDS in 2007 as deputy director, held leadership roles in a number of NIH and NINDS programs, including co-leading the NINDS Brain Initiative. And we're pleased to have John down here, who is the director of the Brain Initiative. John, where are you?
Back here.
Back here. And he's worked on the blueprint for neuroscience, a traumatic brain injury center collaboration between the NIH, Intramural, and the Uniform Health Sciences Services University, the HEAL Initiative, the Common Fund's Undiagnosed Disease and Acute and Chronic Brain Transition, and he was instrumental in founding the NIH Office of Emergency Care Research. And he's just a wonderful and warm human being as well.
Thank you.
Walter, it's all yours. Take it away.
Well, thank you, Mark, and thank you, Congressman, and thanks everybody for coming. We really enjoy seeing people kind of in our tribe trying to work to advance the shared mission that we all have. And we have a great session tonight, this afternoon, this afternoon. And so I want our folks to introduce themselves, get a little brief summary of kind of work they've been doing in this space. And then I'm going to kind of talk a little bit about NINDS in 75 years in about 7.5 minutes. And then we're going to have a really good discussion, trying to hone in on, what NINDS's, place in the ecosystem has been for many different areas of neuroscience, and also get suggestions about what we can do better in the future. So it's going to be a great session, and thanks everybody for coming. Think about your questions. We're going to have a lot of time to entertain them. So with that, I want to pass on to Eileen Villa, who may be the most important person on this group.
No pressure. So I just want to first say thank you. I want to thank the Congressman. I want to thank American Brain Coalition and all the other co-sponsors, but especially Dr. Korshitz, Dr. Kadori, and all of the hardworking scientists that are at NINDS and our other NIH agencies. We owe you such a huge debt of gratitude. We are so grateful, and I know I speak for everyone in this room by saying that. My name is Eileen Penn Miller, and I'm a caregiver, okay? I'm a mom and I'm a daughter. I have a mother that was diagnosed with depression before I was born and Alzheimer's recently. I have two sons, one who's living with bipolar and another who's got a rare epilepsy diagnosis. I have a father who's a Vietnam vet, and he has lifelong anxiety and PTSD. And truthfully, I myself had a really bad bout of vertigo for about 2 years, and inexplicable. So I know what it's like to be one of the 200 million Americans that are living with some sort of brain disorder. And I'm going to take a risk here and just ask by a show of hands, how many people here have or know someone with a brain disorder? Okay. That's about right, because it's 1 in 3 Americans, so there were a lot of hands that just went up. I'm really proud to have been an advocate in this space for the last 20 years, fighting for the epilepsies. We recently had a bipartisan national plan introduced, and I've been working in the trenches for the rare, catastrophic, and oftentimes deadly epilepsies. And it's truly my pleasure to try to represent the lived experience for all the Americans living with these disorders.
Excuse me, can we turn the microphone?
Oh, I'm sorry.
No, it's okay. We got it. Thank you.
I'm Rick Hugan here. I'm the chair of neuroscience at Johns Hopkins University, and been a professor there for many, many years. And I want to thank the Brain Coalition and the Congressman and for all of you coming. And I want to single out NINDS though, because I look back and I've been funded by NINDS for 36 years, so almost half of the 75. So 1990 was my first NINDS grant, and I've been funded essentially through NINDS since then. The what I wanted to get across today is the role of basic science in translation. And I'm a basic scientist. I'm a PhD. I've worked on learning and memory, trying to understand what changes in our brains when you learn something. And so I'm doing biochemistry, molecular biology, and NINDS funded this work. They funded basic biology. And so we study synapses, we're part of the brain, and we characterize a lot of genes and proteins that are involved in learning and memory in animal models, with some rats and mice. And discovered genes that that impact learning and memory. So in 1998, so this is a long time ago, we identified a gene called SYNGAP1. This is a protein that's in synapses. It's really important in mice for learning. So we knocked it out using genetic methods. CRISPR wasn't invented yet, but there's other methods. And these mice had learning differences. They have epilepsy, we now know. They have lots of behavioral problems. They have repetitive features and repetitive behaviors. They, at the sort of cellular level, they have lacking in neuronal plasticity. And so that was really interesting. That but it got more interesting when, in 2009, 3 kids were found with damaging mutations in SYNGAP1. And so, so it turned out that these kids have intellectual disability, they have seizures, they have autistic repetitive features, very similar to the mouse model. So the mice model these kids extremely well. So animal models are really important. And then, so we've been since then interacting with the patients and the families. I'm on an advisory board with a couple of people here in the audience, and we are working with biotech companies to develop therapies to genetically modify and rescue the kids. So this is a great example where basic science. We've worked on this gene for 20 years, and so we had so much knowledge. Ed might talk about this as well. That biotech has really been able to understand the biology of SYNGAP and design ways to try to regulate and cure these kids. So thank you.
Thank you, Rick, for coming.
And I look forward to continue being funded by
Which has that?
Well, I also will start with thanks, and I'm going to switch up this microphone. Let's see, this one must be on already. I'm Erica Augustine. I'm a child neurologist at the Kennedy Krieger Institute, where I'm the Associate Chief Science Officer. And I'm Associate Professor of Neurology at Johns Hopkins Medicine. It's really a delight to be here to celebrate the NINDS 75 years and to join really phenomenal colleagues in this space. I think what you will hear and what you have heard are so many threads that bring us together. You started with the internet, space, and neuroscience, which are fascinating fields that are all very forward-looking and important fields. And when it comes to neuroscience, neurological disorders are the leading cause of morbidity and mortality in the world. Morbidity meaning illness, impact, disability. So neuroscience, at its foundation, really helps us understand how the nervous system works. It helps us understand human disease. And my interests are in how we translate that into impactful therapies. I also have interest in rare disease. So like neurological diseases that affect many, many people, rare diseases collectively are also common. Maybe 300 million people in the world are impacted by rare disease or more than that. And my specific rare disease interest is in a group of disorders called the neuronal ceroid lipofuscinoses. We just say NCLs or back diseases for short. And these are 13 different genetic disorders that share common symptoms under the microscope. They have some common features cellularly, but they cause devastating symptoms: blindness, epilepsy, dementia like Alzheimer's, movement problems like Parkinsonism or ataxia, and bring all those symptoms together and have them happen in an infant, a child, or a young adult whose lifespan is shortened by these severe symptoms. And those are the NCLs. So I'm really compelled by being able to work with families who come into my office and first ask, but what does my child have? What is causing these symptoms? I really want to be able to answer that fully and accurately. The very next question is what's going to happen? As a physician, I need to have a strong scientific understanding of disease course. And then the very next question is, well, what can we do about it? And that's what really compels me and drives me to understand how we take discoveries in the lab and move those along a path that leads us to treatment. And that's the work that I'm interested in. And the NINDS has been there at every step of the way. So I came out of residency, I was interested in learning about therapeutics, and I was really fortunate to have the opportunity to be a part of a research training program at the University of Rochester focused on experimental neurotherapeutics. I learned about how to evaluate discoveries in the lab, to think about whether they were suitable for use in people. I learned about how to design clinical trials. I learned about how to move them forward and continue that support from the NINDS with the Career Development Award, the Neurological Sciences Academic Development Award, the NSADA. And that gave me the time and space to take that knowledge, develop my own research ideas, and those led to my own independent research grants from the NINDS and from other federal sources. And so now we have much better understanding of some of these conditions, particularly one or two, and we are poised and ready to launch how we understand disease, how we measure disease for clinical trials, and how we actually innovatively and efficiently lead to new trials that are going to lead us to new therapies. And so, again, the NINDS has been there at every step of the way, and patients, families, and advocacy partners have been critical. So it's just an absolute pleasure to be here today.
Well, good afternoon, everyone. My name is Edward Kay, and I have the experience of spending almost 20 years in academia. I was trained as a child neurologist and then as a biochemical geneticist. And one of my frustrations as a treating physician was I would see all these rare genetic diseases. I had one of the first neurogenetics clinics in Boston. Didn't know what to do with them. I'd make a diagnosis, and then the family would ask, well, what can we do? And I'd have to say, you don't have a treatment. And so what I was able to do was to really transition into the biotech space. Now, I was able to transition because, very similar to what you heard from Erica, I had a 5-year KOA award, which allowed me to learn neurogenetics. I was trained as a glycolipid biochemist because I was focused on the glycogen storage diseases and the glycolipid storage diseases. But then I realized that the future is in neurogenetics, and I was not aware of everything that was needed. So I wrote the grant, spent 5 years in the lab. That's where I met Walter back in the early 90s. And that NIH funding and the continuous funding afterwards allowed me to use that information. And then when I went into the biotech space, one of the things that I realized I was good at, I could pick out what were some of the assets coming out of really good academic centers. And we had worked very closely with Duke and Mount Sinai. We turned those interesting ideas into therapies for children. And so in my career, I have overseen the approval of 7 different drugs in a little over 20 years. And again, they were all first in class. No other therapies for Duchenne muscular dystrophy, for Fabry disease. We did it for MPS I. We did it for a lot of different diseases. And one of the things I appreciated is we needed that infrastructure that was coming from the NIH and all the funding, the NINDS, that allowed us to figure out is this good enough to turn into a therapy? It was only recently that I met Richard, but I'm a beneficiary of his lab. We use his mouse model to look at SYNGAP1. We think very near to a therapy for SYNGAP1. And again, we couldn't have done it without that collaboration, without that funding that came from the NINDS. And I think one of the things that is frustrating is this is not the time to reduce funding at the NIH. This is considered one of the halcyon days of neuroscience. We are so close to so many interesting ideas. And if we could nibble away at Alzheimer's disease, at Parkinson's disease, one of the things that was very gratifying for me at Stroke Therapeutics, we're working on Dravet syndrome, one of the most common developmental epileptic encephalopathy, very similar to what SYNGAP1. We were hoping that by addressing the underlying genetic cause, and again, until the the NIH funding and the NINDS funding found the gene, we had no idea how to address the underlying cause. And one of the frustrations was we kept on treating patients, these children, with therapies like anti-epileptics, but we couldn't get at the underlying cause. And yeah, we can make the seizures a little better, but not cure it. And we couldn't do anything for all of the cognitive and behavioral problems. And what we're seeing now, and we have this at the clinic, we're in phase 3, we're actually seeing not only a 90% reduction in seizures, we're seeing improvements in cognition and behavior. These are in children who are severely disabled, even in teenagers. 10 years ago, if you had said, was this possible, I would have said no. But now we know Dravet syndrome, Rett syndrome, Angelman syndrome. We are seeing improvements, especially in disorders of synaptic connections, because the neurons are still there. They just can't talk to each other. And so it is so critical for us to continue. The biotech ecosystem is so fragile. And I always think of it as 4 pillars. One major pillar is the NIH that has all the idea generators that you turn into a therapy. And then you have to have the FDA. And we know that the FDA has been adversely affected by a lot of these cuts. And then we have the patient community. The patients are the ones that are driving us. And they're saying, you have to come up with a therapy. This isn't good enough. What are you going to do? This is what we need. And then finally, we also need investors. And right now, investors are absolutely afraid to invest in anything because they have no idea what the economy is going to do. They have no idea what the tariffs are going to do. So we have these 4 pillars, and all you need to do is take one of these legs out, and everything collapses. And we have spent 60 years trying to develop basically an ecosystem in biotech that is so far ahead of everybody else in the world. Do we really want to give it up to the Chinese or to the Europeans? And I would think not. So I'm very pleased that we have this many people that think this is important. But it's so important for us to continue the funding. That basic engine is the NINDS, is the NIH. We need to support that. Otherwise, we are not going to come up with the next generation of therapies.
Well, thanks so much, guys. And as you can see, there's a real string here. First, never underestimate the power of mom. Number 2, it's this fundamental understanding of how the brain and the nervous system works that allows you to jump to treatments that Ed has worked to bring into commercialization through work that people like Erica have done in their clinical trials. So it's a nice string of things to discuss as we get to the questions. I wanted to basically talk a little bit about the Institute, and first of all by thanking the American Brain Coalition so much for putting this on. Let's give them a hand of applause. My best friend Katie Sale and Jenny French. You guys have just been fantastic. As everybody knows, this is our 75th anniversary. Interestingly, NINDS, if you look back at its origin, it was driven by people who really wanted research in neurological diseases, and they just pounded the pavement at Congress until they got the Institute established in 1950, and President Truman signed that declaration to make it happen. A lot has happened over those years. I think Ed and I, as maybe the senior neurologist here, remember, as he mentioned, when we trained, neurology was a diagnose and adios specialty. The thing was, neurologists, they were really interesting, they were totally useless. But that really changed over the years. And that changed a lot because of this explosion of knowledge that moved into neuroscience that then got taken up by all sorts of folks in this ecosystem. And when you think about NINDS, most people think of it as a government agency, but really it's a community. We have staff, and we have staff here who work really hard. I want them to stand up and be recognized. Can the NINDS staff stand up, please? Come on. Staff that are working to move grants out, and most of the ecosystem we deal with are the grantees, and they're in every state in the union, multiple universities, and we run biotech companies to get them started, to move into, get therapies for our disorders. We have an intramural program. I saw Catherine Roche here. Catherine, say hi. From the intramural investigators in Bethesda, and the ecosystem is the grantees, the staff, and the patients, and the people who have the disorders, and the people who advocate for those people who have the disorders. And NINDS really wants to cultivate, and we have over the years, as I've known it, an incredibly friendly, open door policy to anybody who wants to come in and discuss things with us. We have, as our MO is to advance our understanding of the nervous system, and to use that knowledge to get better treatments for diseases. And we'll listen to anybody who has good ideas, and we have certainly engaged in lots of public-private partnerships over the years to make things happen, because we know that by ourselves, there's limited things that we can do, but as an ecosystem, there's really nothing that we can't do. So, you know, in the years that I've been here, you think back, you know, when I came out of training, I think we had 3 drugs for epilepsy, and now, I don't know, it's like 28 or so. Most of those actually came through the epilepsy screening program that NIH runs in Utah, where people come in for free and screen their drugs to see if they work in epilepsy. Stroke, which was my field for a period of time, people would come in with a massive occlusion, stroke, and there was nothing that anyone could do for them. They would be trying to prevent pneumonia and move them to a nursing home where they would spend the rest of their lives. Now, and this is the work that I got involved with, we can, and it's routine now across the country, you can put a catheter in and you get the clot out, and if you do it in time, the person can do extremely well. I mean, Senator Fetterman is, I think, a good example of that. But there's lots of examples that every day in this country, stroke rates in the United States have decreased by 70% since I started in medical school. Unfortunately, they started to go up in the last couple of years. We have just a reminder that you can't give up. You got to keep pushing on this. But if you think about a 70% reduction in stroke rate, how many people escape a stroke? They don't know that NIH had anything to do with it, but NIH did trials that proved, you know, say that anticoagulation prevented these massive strokes due to atrial fibrillation in at least half to 75% of people. So over the years, there's a lot of prevention that we've done, and millions of people have really escaped these conditions. But on the other hand, we know that there's a lot more we need to do. We have lots of drugs for epilepsy, but still 30% of people have epilepsy that's uncontrolled, right? 30%. Stroke, if we get people in time, but you know, the majority of people show up and it's too late. So we have work to do there for ischemic stroke. For other types of stroke, we don't have those treatments. We have this miracle that happened in spinal muscular atrophy, where an antisense molecule, kind of similar to what Edmund's working on, was able to change the splicing abnormality, make a splicing change in people with spinal muscular atrophy. And there was a clinical trial, and halfway through the trial, everybody knew that it worked because it was like a miracle. It wasn't like they got a little bit better, they were like really better. So kids that would have been dead at a year survived, were walking, not on ventilators. And now, you know, we have that as a success, but then we have how many other? We have 400 other neurogenic diseases, and they can say, why don't I have that therapy? And there's not a really good answer for that. It's really a responsibility now that we broke this ice, that we got to really move hard to get these type of therapies out and to treat more of these kids with these different neurogenic disorders, and that's what you heard a lot from today. Multiple sclerosis. Also, when I came out of medical school, we had with steroids and really bad cases, Cytoxan, the chemotherapy drug. now there's at least 16 disease-modifying therapies for exacerbating remission of MS, and they're very powerful. We have treatments for Friedreich's ataxia recently, Duchenne's muscular dystrophy, and then Parkinson's disease, as you know. Some of the old therapies with Cinemet are still actually probably the best medications. But we have deep brain stimulation that is very effective in a number of people. And we have groups working in the Brain Initiative to understand the surface of the brain, to manipulate them like we had with DBS for Parkinson's, but in other diseases, including, where's Mark? Depression, post-traumatic stress disorder, obsessive compulsive disorder. So the neurologist will soon be taking over psychiatry.
It's all one brain.
All one brain, that's right. But yeah, that's how progress is made. The Brain Initiative is just like, it's just unbelievable. I mean, the congressman spoke about space. I would just say that when we had the first meeting to do the cell census, the first speaker was a cosmologist. And so he was invited in by Jim Eberwein at Penn, and we were sitting there listening to this guy talking about how they look at the cosmos and the different stars, and I was like, what is going on? And then he said, and it's just like what you have to do. You have to be able to use this data and distinguish these different cells, just like we're doing with the stars. And then he went into these, mathematical equations that we're going to have to use, that he used on the stars, and it all kind of made sense. So space is not, there's something there. We've also had the real opportunity to, the beneficiaries of targeted funds from Congress to do things like the Brain Initiative is clearly the biggest thing we've done. And John Nye has been amazing running it. And we also have the HEAL Initiative, helping to end addiction long-term, working with NIDA. And our job is to try and find non-addictive treatments for pain, because if you're going to get rid of addiction, you got to get rid of opioids, and we need better non-addictive medicines. We work hard on the national plan for Alzheimer's disease, particularly in, we call it Alzheimer-related dementias, frontal temporal dementia, Lewy body dementia, vascular cognitive impairment. And the truth of the matter is, most of the elderly people they have a diagnosis of Alzheimer's, but they don't just have Alzheimer's. They have a mix of these other pathologies. So what we've been doing for all these years is really going to be super important as we move into treatments for Alzheimer's disease. I think, the future, we kind of put this group together because one of the things we are putting our investment in is genomic therapies. We can see that these neurogenetic disorders are treatable, that it's doable, that we can scale it up, that we can work, the academicians around the country can develop treatments in their labs, which they couldn't do if you're looking for a treatment with a small molecule. It would take 10 years for the big pharma to do that. But these treatments are different. These are really scalable once we get the roadmap established. So we're trying to make a big input in this space, particularly in the neurogenic disease, because they have no other treatments. But these treatments, ASOs, interference RNAs, gene editing, gene replacement, they are precision therapies that are going to be incredibly valuable as we move into the common disorders once we provide evidence of efficacy, how you do it, and safety in these rare disorders. So you can think about Alzheimer's disease. Biggest risk factor for Alzheimer's disease is ApoE4. You can edit ApoE4. In an animal, you can edit it, and you can make a big difference. So we could go from genetics in neurogenic disorders to common disorders pretty quickly once we get our feet planted securely in a safe environment. So I think I'm real excited about the future of neuroscience. I just checked today, 2024 data. Neuroscience is getting a little over $11 billion at NIH, and that's more than cancer, it's more than cerebral vascular disease, more than infectious disease. It's the most of any area of science. And that's because people are intrigued by the brain, the nervous system, and its disorders. and they're going into it, and really smart people are going into it, and they're writing great grants, and they're getting money. And that's how the system works here at NIH. NIH and NINDS is a genuine American system. Everybody has a chance, put their ideas on the table, get reviewed by your peers, and if you get a good score, you get funding from the government. You go around the country, around other different countries, it's not exactly as democratic as that. It's more who you know, what was your position. Here, it's really wide open, and it's a great place for people to come and explore their ideas. It's not an easy business, as Rick and everybody can say. We only fund the top 8% of the grants now, so that means 92% we don't get funded. And that's just because we fund the best until we run out of money, and now we're running out of money at 8% of the best grants. So there's a lot more out there that we could be doing, and we're always at NINDS trying to innovate, trying to do something creative, improve the ecosystem, which gets me back to the beginning again, which is you all are the ecosystem. You represent the bigger ecosystem of the people with disorders, the people who advocate for disorders, the grantees, the scientists, and we really appreciate the privilege of working with you on these, what I call just inherently important problems for people. So that's kind of my quick summary of NINDS, and that's my story. I'm sticking to it.
We were taught that 50% of epilepsy was idiopathic. In other words, we didn't know what it was caused by. And I remember we would, as residents, we would say, yeah, idiopathic means the doctor's an idiot, the patient has a pathology, because we don't know what it means. And that we now know that 50% is all genetic. And so we have such an understanding, and that didn't come out of the air. That came from hard work that people like Richard were working on and talking about neurotransmitters. So every time we learn something about a rare disease, that knowledge can be expanded into other indications. We're learning that. Another example, we have a rare condition for ophthalmology, autosomal dominant optic atrophy, and basically, which is another neurologic disease, a disease of the optic nerve. The cause of it is due to a protein that's important for mitochondrial function. Well, we can actually increase ATP, which is the energy source. And so what we're doing with that, we're going to say, well, gee, why can't we use this for Alzheimer's disease or Parkinson's? Because that's an energy deficient disease. And so sometimes you just come across information and you can realize that a drug can be used for multiple indications. And the only way you know how to do that, and I think, a lot of the advances have come from trying to understand these rare diseases, because you can get down to the molecular level, you can understand the genetics. But one of the things that will be important is in what we're finding out in adults with Dravet syndrome, they've never been tested genetically. So most of the adults who have intractable epilepsy have never had a proper diagnosis. And it's simple, it's a simple blood test, you can do it. But you need to know the underlying disease before you can come up with a treatment. So I think it's really trying to treat the entire patient, and how do you approach it, how do you use this incremental information and then expand it into larger indications.
And Eric, I was going to ask you to fill in on a list that I made, which is, you think about the ecosystem, the people who actually contributed the most were the patients who went in trials. So I want you to talk a little bit to people about what it takes for a family and a child to go through these trials. And I think we really need to recognize that they're usually the heroes that people don't recognize, but really are the heroes.
Absolutely. I mean, clinical trials take such commitment, and there's such a long journey and path. So, we're talking about the discoveries and the advances being made at your company, the genetic basis for that. You can even go back beyond that or before that to the Human Genome Project funded by the NIH that led to really a boom along with other technologies in discovery and identification of genetic causes of rare disease, and those are leading to rare disease trials, and then we get to common diseases. But all along the way are the people who participated in research, who contributed their time and energy, and made a major commitment to clinical trials. So clinical trials are on investigational products, where we learn as much as we can from disease models before we move into people. But they are people who are volunteering to take a risk, potential benefit, but certainly risk as well. So that's the first level of commitment. Then there's a time commitment. And in neurology, that time commitment is often long because the diseases that affect the nervous system are complex. Often they unfold slowly, and so that can mean that the trials can be long. Many visits over many years sometimes, very long periods of follow-up. And think about the last time that you went for a regular doctor's office visit. Was it a quick thing? Was it a half an hour? Was it an hour? Or did you take half a day or a whole day out of work actually just to do the one appointment for the one thing? When it comes to clinical trials, those can be multi-day visits. They are at specialized centers. They're not necessarily everywhere. So the travel that's involved, the time commitment, the risk acceptance, and just the commitment to follow through all the way to the end is incredible. Now, we do a lot to try to mitigate that time investment and the needs. At the same time, our interventions are more accurate and more precise, and they're also more involved to administer. And oftentimes in neurology, those treatments may be directly administered by injection to the eye, or injection to the lower part of the spine, or sometimes injection directly to the brain. And so to be more precise, to be more targeted, again, that takes us really having sometimes more invasive kinds of approaches, but ideally greater yield, greater benefit, greater impact for families as well. But it's a huge commitment to be a part of a clinical trial.
Sue?
Thank you. My name is Susan Dickinson. I would just start by saying I was trained as a genetic counselor, and I've worked with rare diseases for more than 25 years. And one of the really special things that NINDS does so well is incorporate the patient community and their families. Under Walter's leadership, this has really expanded, and every year they host a nonprofit forum, which is tomorrow. If anybody, I'll give a plug if anybody wants to come up to Rockville. But it's important because as you all have been saying, everybody's life is going to be touched by one of these diseases. You may not know it yet, but you will experience it. For the past 18 years, I've been CEO of the Association for Frontotemporal Degeneration. FTD is one of the rarer dementias that Walter mentioned earlier. And our disease is hard to diagnose accurately. We don't have biomarkers. There are no disease-modifying treatments that have been approved for any form of FTD. And today we have 7 clinical trials. And we have a community that is so eager to participate and partner with the companies and the scientists who are running those. And one additional point I would make to, if drastic cuts in funding are realized is, you can turn off funding in an instant, but you cannot, the infrastructure and the work that is lost will take years and years to rebuild again. So cuts like are proposed that may be realized are gonna affect at least a whole generation to come.
I want to pause for a second. I was going to I'm going to tell you guys what the next question is, which is, if you're the director of NINDS, what would you do differently? You'll have time to think about your answer.
So, we're taking a break because we have a special guest. Congressman Mike Thompson represents California's 4th district, which is Napa, basically, right?
Napa's included. Napa, Sonoma, Lake, Solano, and Yolo counties. This is from California.
Half time.
2 people.
So,
There's only 2 people in California.
I know, 2 more here.
I said half the year.
Pardon me?
Half the year. Well, Congressman Thompson joined as co-chair of the Congressional Neuroscience Caucus following the retirement of Representative Earl Blumenauer. And Congressman Thompson brings a long-standing commitment to healthcare, mental health access, and scientific advancement principles closely aligned with the goals of not only the Neuroscience Caucus, but the element of today's discussion. So we thank him for stepping into this leadership role and continuing the caucus's vital work. So let's welcome Congressman Thompson.
Well, thank you very, very much for inviting me. More important, thank you for being here and everything that you do. There's nothing that I can tell you that you don't know more about than I know about. So you know how important this is. You know how these issues touch every one of us, everybody we know, everybody we're related to. You know how much work needs to be done, and you know that we really need to be stepping it up. I came in just to hear at the tail end of your question, and I'm assuming it has to do with the cuts in research. And you're spot on. Those research cuts are going to set us back generations. And it's not just in the neuroscience space, it is every place where research is being done. One of the other things I do before Earl got me involved in this is I chair the task force on gun violence prevention. And we just had a briefing downstairs by professors, medical doctors, and researchers from 5 different universities across the country on they study gun violence prevention. And for years they got nothing in federal funds because there was a law that prohibited any research on gun violence. And we were able to reverse that, and we got them $25 million. Now, you know that in the research space, it's not a lot of money, but they've been doing amazing work, and now it's gone. And they were telling us what's happening. They have professors who they worked with who are just gone. And there's not a lot of places that do things like gun research. There's probably not a lot of places that do some of the research that you all depend on. I know other governments do. I have one researcher who lives in my hometown was fired from UC Davis. She was doing major research, and she was immediately hired by Denmark. So that's a race to the bottom. All of a sudden, Denmark has a better researcher than not only California, but the United States. So you're right, it is just tragic. So my message to you is keep the faith, keep working. I've got your back. I'll do anything I can to make sure that your work is protected and we make the advances necessary to help the people who we all agree are our #1 priority. So thank you very much, and a phone call away.
Okay, so I hope you guys are thinking about your answers. But really, the issue is you all come from different perspectives, and so you can kind of look into the future, and if there's something out there to grab you think is, really the most important thing right now, what would it be for you? Why don't we start with that, and we'll work backwards this way. Give my a break.
One of the things that I saw a lot in academia, and probably Erica saw it too, is there's often really promising physicians who fall in love with research and want to do it. But I think for right now in academic institutions, there's such a push for clinical care and a need, and less and less physicians. So how do you nurture that physician scientist? And I was one, but I have to say it was a tough road. And you had to be very jealous of your time. And so what I get concerned about is we have, and I do think, having been trained as a physician and as a scientist gives you some unique abilities to observe. Because you really do, you understand the pathology, you understand the underlying disease themselves. And then now you can have the ability to understand the science. So how do we make sure that next generation of physician scientists can get the funding that allow them to have a successful career? And obviously it's the same is true for PhDs. We need to make sure that they continue. It's just harder and harder to have a career in any science, including neuroscience. So how do we make sure that these people have the time and the money and the commitment to be able to really grow their career? Because it does take some nurturing to get you to that first NIR1 grant. It's not easy. And then once you get there, hopefully that's sustaining. But we have to do a better job of getting these people to their first R1 grants.
we certainly at NINDS, that's, high priority for us is not mortgaging the future by losing a generation. So everybody, and I'm going to ask, maybe, I had once kind of sidelight to what Ed was saying, maybe we follow up on it, is that one of the ways you can get people involved in research is for them to be adopted by a disease organization. So I think that the advocacy groups should not underestimate the power they have in cultivating those kind of relationships. And I can tell you for myself, I got a grant from Huntington's Foundation to get me going, but the main thing was, they brought me into the fold and I met these people that was just phenomenal, and you would never think of leaving.
There's nothing more compelling than people and relationships, and I think it ties together several threads. People pursue science, physician scientists pursue the science that they do, often because of a family they met, a patient's story, someone they can't get out of their mind, something that is just, I have to change this. I need to make this a different path for someone else. And so, one of the ways that we really engage people to think about science and to think about medicine is to bring them along. Come to clinic, come with me to the nonprofit forum, come with me to an advocacy organization family meeting. And you do that every year. You bring people back. Say, come, what's your idea? What is your scientific idea based on what you saw or what you're thinking about? Okay, let's turn that into a project. Let's turn that into a fundable project. What do we do next? Come back and share the information that you learn with families. And yes, advocacy organizations, family organizations, also those small amounts of seed money, again, they maintain that interest and enable projects to become larger projects. And so it's really very, very important. And a couple too, I want to pick up on your point as well, around the need to fiercely protect and guard those training programs and career development programs. It is how people are able to carve out the time to learn so that they can really pursue rigorous science.
So an example that I had at the Children's Hospital in Philadelphia, we had made a diagnosis, and I was head of biochemical genetics of methylmalonic acidemia, a rare organic acidemia. And the father was a state senator, very engaging guy. And he said, what can I do to advance methylmalonic acidemia research? We had no research. And so he went in and he got his colleagues to give us $50,000 targeted for methylmalonic acidemia. So I had one of the biochemical geneticists, one of the residents, and who wanted to do dysmorphology, which is looking at basically trying to make a diagnosis based on facial features and other characteristics. And I said, no, you need to do biochemical genetics. And I said, I'll give you $50,000, take that money, make a mouse model for methylmalonic acidemia. And he did it. And now he is a senior investigator at the NIH. He has used that model. He has developed gene therapies for methylmalonic acidemia. He's one of the leading researchers. And it was that father who said, there's got to be something I can do. And he said, I'll give you $50,000, which seems like a very small amount. But that was enough, working with one of the senior geneticists at Penn, to make that model. And then he took that model with him to the NIH, and it was very successful.
I would add on the what would I do piece of things as well. It still relates to people. And it's about the exchange of ideas. When you talked about Society for Neuroscience and the thousands and tens of thousands who attend the meeting, it's the public forum, it's the public square for exchange of ideas. It's where you synthesize what you found, you convey that clearly to other people, you put it out there for critique, and you get questioned, you defend your work, and you think about how to do your work differently. With the nonprofit forum, it's an opportunity for advocacy organizations and other nonprofits to learn from one another. And I think in addition to fiercely protecting training programs and early career development programs, bringing people together. I think we think of NINDS primarily as a funder, but NINDS is also a convener. And I would hate to see that lost, because it is the opportunity for the cosmologist to work with the neuroscientist and think about new mathematical approaches. It's the opportunity for the cardiologist and the neurologist to work together on a new project. It's the opportunity to have the basic lab scientist actually work with the clinician scientist to realize, oh wait, actually the approaches you use for measurement people, we actually can do that in mice. We can do that in this way. And so that crosstalk is critically important for the formation of new collaborations and new ideas, and I wouldn't want that to be lost.
So Rick, what do you see looking through the crystal ball, and what would you want us to do?
So it's probably no surprise that once you maintain that, basic science funding. So there's a lot of pressure, people want quick fixes, and you and I know that basic science is so critical for the, where we are. And I said earlier that this is the pinnacle of science. I can't imagine what I'm doing now 10 years ago. Can you imagine what we're going to be doing in 10 years if we keep funding? Because all the techniques that we're developing, especially for genetic disease, but not even rare genetic, extremely ultra rare. There, of course, many of you know the company NLORM, where it's a N of 1 that they're developing therapies for individual mutations in children. And because the techniques have gotten more standardized and can be used very, very quickly to on 2 or 3 people have the same mutation. So I want to call out the BRAIN Initiative, because the BRAIN Initiative really has emphasized tool making and pushing the community to develop these techniques to get us where we are, and it has a huge impact in the progress we've made in the last several years. So I think I'm looking forward to the future, and that's going to come, I think, from continue developing tools, developing a basic science understanding of genetics and these therapies. So there's of course there's CRISPR. Who would have thought that these little enzymes would be transformational in science, and the ability to prime edit now, to actually edit very quickly individual mutations again may become standard. In 10 years it may be just the way we treat genetic and even the extremely rare genetic diseases. So keep up with the basic science.
Absolutely. Eileen?
I think just to maybe riff on a few things that were previously said, I also want to commend the nonprofit forum is one of my favorite meetings, because those of us that are maybe steeped and siloed oftentimes in our own disorders, whether it's epilepsy or Alzheimer's or any of the other ones that were mentioned today, we get an opportunity to learn from each other. So I think this role of NINDS as a convener is incredibly important. I also think that working through sort of the science from the basic through the translation and the clinic through to the integration, right? Like, how do we make sure that everybody has access to these advantages, and also the population and the surveillance? And I think NINDS has done a good job of being a partner with other agencies, both within and outside of NIH, but sort of looking forward, I would like to see even more of that, because we've got to understand how many people have these conditions. We have to understand what causes them in order to be able to realize those precision and other therapies. And then I would also just say, I think the interagency collaborations, NINDS again, has really been a leader through BRAIN and through other initiatives, working with NCATS, working with CDC, working with other NIH institutes. Our diseases, epilepsy, yes, it is a brain disorder. Yes, it causes seizures. But the community of the rare epilepsies that I run in, they are nonverbal, they are not mobile, they have respiratory conditions, gastro conditions, musculoskeletal conditions, so it's every agency. So I would ask you and the other institute directors to really think about how can we identify those opportunities for collaboration so that we can redress the whole person. Because it's the whole person that's managing this disease in each of our instances. And I know it's true for epilepsy, but my suspicion is that it's true for many of these other disorders as well.
You know, I think that's absolutely true. I told Dr. Collins years ago, we need a new institute for things that fall between the cracks. Trish?
You know what I'm gonna say.
Yeah.
Okay. Obviously
Want to take
funding,
that? Okay.
now I can talk loud enough. Obviously, the funding is critical right now and the highest priority because of the situation that we're in. However, someone asked, what can we do in addition to talking to people, Congress people, everybody next to you? Make plans now to donate your brain when you die. There is no more precious resource to neuroscience than donated human brain tissue. Had to say it, you knew I was gonna.
So this, why don't you tell the backstory of how you guys into this?
Okay. My father died of Lewy body dementia about 10 years ago. We donated his brain. It was an unsophisticated platform at the time to do that. However, we did it. It almost fell apart at the last minute, and then being a former journalist, I wasn't afraid to call people. So I called this guy named Walter Korshes, and I said, what can I do to help you get more brains now that I understand how important it is? Can I help you? And he said, yeah. So they got this thing called the Neurobiobank, which supplies donated human brain tissue to researchers all around the world. All around the world. If you are an investigator and you need tissue for your study, and everybody needs to validate at some point on human brain tissue, you go into their portal, you enter the specifics of your disease, you look through what is now a collection of pushing 18,000 brains, you make a request, you get an answer, and all you pay is shipping anywhere on the planet. Any neuroscientist will tell you this is an incredibly precious resource. There is no substitute. So please consider doing that. You know, if everybody does a little bit of a lot of things about funding and talking and these things, we're gonna make a difference. Thank you.
The best phone call I ever did.
Thank you, NINDS. I am Jill Wood. I am the CEO of Phoenix Nest, and I'm also the parent of a child with MPS 3C and ultra-rare neurodegenerative disease. And we're all sitting here preaching to the choir. I think every single one of us is wondering how the hell we got here. And we're about ready to lose 40% of our funding. My company,
Okay.
yeah, and my company, Phoenix Nest, survives off of NINDS grants. We have, one of our programs has 5 patients in the United States, okay? And we're this close to having a treatment for our enzyme replacement therapy for MPS 3D, as well as our gene therapy. But so what I wanted to say here to everybody that has won one of these grants is that we need to really give the NIH props. We need to advertise for the NIH. They do not go out and commercialize them themselves. The average taxpayer does not know what the NIH does. They don't even know what it stands for. So I think we should go back to our websites. We need to cue it in. This funding to start this research started with the NIH. And I think patient organizations on your websites and during your social media, this research started with NIH funding. Even if it didn't come to you, it came to your investigator. Because those academics, they don't have the opportunity to go and brag, hey, we got this NIH funding. That's fine.
Well, that's the best ending I could ever. I want to thank everyone. Thanks for the
So, thank you, Paul, for speaking to us. Walter, thanks for putting us together. And thanks to all of you for coming here. It's a great run. This is one of the greatest crowds I've ever seen. So, in addition to thanking the panel, thank conference. We're grateful to our supporting organizations because this wouldn't have happened if it weren't for all those organizations I named at the beginning. Now, it's also important that we call on Congress and that all of you participate, I'm going to reiterate it, that we are faced with an existential threat to not only to biomedical research, but to those people who suffer from broken brains and to those people who will provide a pipeline to carry on when those old folks finally give up. Okay, this is critical. And please call out to Congress, call out to everyone you know to say that we can't let this happen. NIH is really the cornerstones of our biomedical research enterprise. It's really made, as you heard today, life-changing discoveries which have spun out into companies, which have spun out into therapies for people. It's one seamless garment that is now being threatened to be ripped apart at the seams. My dad was a tailor, so I know about that. And we need to work to continue to improve the lives of patients. So we're also really concerned about current legislation that represents a haphazard and ill thought out approach to consolidating NIH institutes in a manner that completely reverses brain and behavior as if they are as if they are not interdependent. And again, this is something we have to be concerned about and something we have to speak out against. And we're also worried about training and the fact that it is so important that young people be attracted to science rather than discouraged from going into it. And gee, that's not a very fun note to end on, is it? But you know what? Let's keep up the fight. Let's all go out and do what we can. Let's celebrate these advances and make sure that they continue. Thank you very much.